Hello?
Can you hear me?
Yeah, I can hear you.
Oh,
Yeah.
good.
Yeah.
My camera is attached to the same thing as my microphone, so unless I want to put my earpiece in and do Bluetooth, I don't get to do visuals.
Oh, okay. I see. Okay.
It's odd.
Yeah.
My Bluetooth was going dead one day, and I was talking to my advisor, and I said, "Do you mind if I, turn off my visuals?" And he said, "Well, okay." And so I did, and I said, "That's because my Bluetooth is going." And he thought it was weird.
Yeah.
It's okay. I've got a strange computer.
Oh.
No internal camera.
Yeah. Is that a laptop or just
It's a laptop, and it's a gamer's computer.
Oh, okay.
So it's faster, but it doesn't have enough C drive for me, so I'm continuously fighting with it and transferring files to the D drive and then being scared to erase them on the C drive unless I've double checked that they're on the D drive. And the whole thing is like not nice.
Yeah.
So I erased Mathematica from my C drive. I haven't afforded it this term, so I just erased it, and that freed up a whole bunch of space.
Yeah, it usually does.
Yeah, it's quite heavy, that program. I'm surprised. I think it was 10 gigabytes or something. It's just huge.
Huh. Yeah.
Anyway, I guess I want you to have a safe election down there.
Thanks.
Yeah.
Yeah.
I don't understand U.S. politics. It's just kind of flies above my head, and I go, "What?"
Yeah.
I don't understand. So I just wish everybody a safe election.
Yeah, thanks. I don't know if anyone else is going to join us today. I know there's a time change this weekend, so.
But that's okay. My body thinks that it's later in the day, so that's good.
Yeah, it's yeah. So I mean, and I made a mistake in the email. I said 2 p.m. UTC, which is, of course, because I think it only changes for certain parts of the world, not others. But it's like everyone eventually makes the change to the same time, I think. So it's there's a couple weeks in there. It's usually pretty chaotic, where no one knows it.
Oh.
You have to look up the time constantly, and so it should be. I sent a correction out last night. It should be 3 p.m. UTC because, of course, North America moves up an hour, but like I don't think Europe does. But anyways.
So somebody might turn up in an hour.
Well, we've had that problem in the past. So people show up. Oh, I thought it was an hour later. Oh yeah.
And I'm really glad that I was able to attend Neuromatch and that I heard about it through this group here. Because I have a lovely paper, and it's called, let's see, Mechanical Surface Waves Accompanying Action Potential Propagation.
Okay.
So it's another waves paper, but it's about surface waves on neurons, when they're actually propagating surface waves. So I wonder how myelin sheath plays into that one.
Yeah. Yeah, there is this something that you've published in the past, or is this a working paper?
Oh no, this is a paper that my match on Neuromatch sent me.
Oh, okay.
His name is Daniel.
All
And
right.
the only one who contacted me, and then I wasn't too sure about going about how to go about the other ones, so I didn't do what I was supposed to do and contact my contacts except for the one.
Oh yeah, yeah. They send you these matches, and like you can contact them, or not. I gotta contact mine yet still, but it's like
Oh, okay.
yeah, it's just an opportunity for you to like talk to people, meet people, new people you might not meet otherwise, and it's pretty fruitful. You can have a discussion about different things, and that sounds like an interesting paper. Um, Certainly,
Do you
maybe we
want me
can.
to put its title up on the chat?
Yeah, go ahead.
Okay, I'll just find it here and
All right. So I'm gonna start the meeting. Welcome to the meeting. Um,
hi.
well, you can put it in the chat. I'm just doing this for
Okay.
people on YouTube. So if you're viewing this
All right.
recording, welcome. We have a couple of things to talk about today. So I want to go through some things because we haven't had a meeting. We had a meeting last week. It was sort of informal, but I wanted to go through some things we didn't talk about last week. So let's see. First thing I want to talk about is I wanted to recap Neuromatch. So we we had a presentation, a couple presentations representing the group at Neuromatch. We had talk with myself and Krishna Katyal on the contrast between biological neural networks and artificial neural networks, and that was actually pretty well received. People enjoyed that. It was a, a collection of different thoughts about the differences between artificial neural networks and biological neural networks, specifically like the kinds of things that artificial neural networks don't have that biological neural networks do. So we know that like biological neural networks, something you'd find in an organism, a network of a dense reticulating network of neurons, of course, can do a lot of amazing things. And while artificial neural networks can do those too, there's obviously a gap. And so what is that gap consist of? And so we've got a couple of candidates in the talk. Okay. And one of the more interesting ones, I think, was where we talked about the sort of the scale free nature of a biological neural network. That is where you have processing hubs in different parts of the brain or the network, and those are both for information processing purposes and for energetic purposes very efficient. And so your average artificial neural network is very there's sort of a random connectivity, and then the weights are shifted so that you can find patterns and learn patterns, but there isn't this general design principle of sort of a hierarchical set of modules that do things and communicate through these hubs. And so that's the sort of thing that if you know anything about biological neural networks and sort of the literature on um, the literature on complexity theory or on some of the uh, brain networks, that's out in the literature, then that's kind of obvious. But to think about that in terms of an artificial neural network is maybe something people don't do very often. And so Susan put her link in the chat here. So this is mechanical surface waves accompany action potential propagation, and this is a Nature paper from 2015. And this is open source, so it's available for free. I don't know if we can get into let me open a window here. Okay, so this is actually, let me share my screen. Why don't we look at the paper? So there we go. So this is the paper here. This is the ResearchGate version of it. So yeah, many studies have shown that a mechanical displacement of the axonal membrane accompanies the electrical pulse, defining the action potential. Despite a large and diverse body of experimental evidence, there is no theoretical consensus either for the physical basis of this mechanical wave, nor its interdependence with the electrical signal. In this manuscript, we present a model for these mechanical displacements as arising from the driving of surface wave modes, in which potential energy is stored in elastic properties of the neuronal membrane and cytoskeleton, while kinetic energy is carried by the axonoplasmic fluid. In our model, these surface waves are driven by the traveling wave of electrical depolarization that characterizes the AP, altering the compressive electrostatic forces across the membrane as it passes. Okay, and then the driving leads to co-propagating mechanical displacements, which we term action waves. And so these action waves, so this is in an adult nervous system, I take it. So it looks like that's what it is.
I don't even know what species.
Oh, they say that they're looking at results from garfish olfactory nerve and squid giant axon.
Oh, okay. Yeah, squid giant axon. It gets quite a bit of press.
Yeah, it's because
Easy to look at.
it's so big, like physically,
Yeah.
that they can see. I mean, it's not that big, but it's
similar to studying axolotls for embryology. It's huge.
Yeah, definitely. Yeah. So it's that's good. Thank you for that paper. PDF available, so I guess you can download the text. Oh, you have to log in. I'm not going to log in right now, but oh, there it is. Okay, so it downloaded it. Okay, so that's that paper. Thank you for the link. Okay, I don't want to do that yet. The other thing that we had at Neuromatch was this Psychophysics of Non-neuronal Cognition talk, and this was on Thursday. This was a group effort, so this is the people who have been talking about this topic in this group. So Dick Gordon, Thomas Harbeck, Jesse Parent, Asmit Singh, and Ujwal Singh, and then Akshara Gopi, who's part of our other group, and myself. And so, that my philosophy on authorship is pretty broad. It's basically if you contribute something to the discussion or the, putting together the slideshow or whatever, or if there's a paper contributing to the paper in some way, then you get an authorship credit. So it's much more liberal than some other people, have authorship criterion in some groups is like you have to be like really close to the, you have to be basically doing a lot of work on the paper. I'm not really that worried about that in these groups because we have so many ideas floating around that it's hard to know. If Susan, if you want to be part of this, when we write the paper for this, we can add you as an author. I'm sure there'll be something that you can contribute to on this. But
okay, if you can find something that goes with what I know.
Oh yeah.
I'm sort of, I don't know, biomedical engineering, electrical engineering. Yeah, so I would have to fit in with some of that I know.
Okay, yeah. So that's so this is I'm going to go through the presentation here because I don't think anyone here has seen it. Okay, so the first thing is we had a couple of short videos that I clipped from some of the data that we have. So this is basilaria in the wild, and I know maybe some of you haven't seen this. Okay, gotta restart this. This is a weird thing in Google Slides, is that you have to restart it to play movies. But this is in the wild. This is kind of like a couple of colonies that aren't isolated, and they're making all these movements. They're shifting around in different directions, and you see the cell colonies, and the colonies are kind of moving they're not even like an accordion like you'll see in the next video, but they're kind of just moving around like, I don't know, almost like if you were dragging some straws across the table that were kind of stuck together with, in some way. They're really, yeah. So they exhibit these kind of behaviors in the wild, and you can see they're moving in different orientations, and they're moving back and forth and undulating, and you see these stacks start to unfold. And then
what
magnification
is that?
Oh, I don't remember. I think it's like 10x or 20x. I'm not really sure. But this I think is 20x. So this is the isolated colony. And so this is where it's like just on a flat focal plane, and you can see that now it's doing this sort of accordion-like movement. You have a leading cell here, you have a stationary cell here, and you can see it sliding. And you can see different modes of movement. You can see these cells are kind of bunching up and then stretching out as they move along. And that'll be important later in this talk to understand. Oh. Okay. All right. I guess we're okay now. Okay, so then we're thinking about this in terms of psychophysics. So non-neuronal cognition, and I'll get into what that is in a minute. But psychophysics is this branch of psychology, branch of sort of physiology that deals with how people perceive stimuli and, encode it, and then you can measure it using some sort of mathematical model. So there's a whole literature on this in humans. Of course, you can see someone in a virtual environment. People have also observed psychophysical properties in mouse visual systems. So this is a mouse focusing on a cat and then focusing on another mouse. You see in goldfish, there's been work done on, I think, color vision. So they've done a lot of, they've sort of extracted a lot of psychophysical laws in goldfish. And then the question is can we also observe things like this in diatoms? And so, it's kind of a stretch maybe, but I wanted to establish that it's something that we see in animals, non-human animals, humans, and then maybe even diatoms, who are animals but they're eukaryotes. We'll get into that in a minute. So this is a short introduction to diatoms. So this is the slide that I used for the short introduction. So they're algae. So they're single-cell eukaryotes, but they're not really animals. They have plant-like characteristics. So they have chlorophyll elements and a silica cell wall. So it's very much unlike animals, but they do have a lot of sensory behaviors, much like plants, but they're not, they're sort of on the border. They're sort of an early branch of evolution. So you have these diatoms. They're glass. They're silica cell walls, and they come in a bunch of different shapes. And so in this talk, we'll be focusing on this Diatom. So this Raphid class of diatom is, you have a bunch of, properties of the cell. You have valves, and you have these girdle bands, and you have all these properties. So just keep that in mind in terms of the morphology. And then you have all these different shapes that they can achieve. So rod shapes, flake shapes, 3D shapes. You can see there's a diversity of shapes. But all of these cells are only at about the size of 10 to 80 microns. So it's a very small time scale. So needless to say, they don't really have nervous systems because there's no room for neurons in this system. You just have a bunch of these Raphid diatom cells, at least for the genus we're going to be talking about. And so this is an analysis, again, of bacillarian culture. So this is, again, a repeat of the video that I showed before. And notice a couple things. One is that you have this accordion-like movement. And we can model this as a series of oscillators. So there are no neurons here, but it's exhibiting some sort of complex behavior, collective behavior. And the question is, what is this behavior? Is it generated by, it's not generated by a brain, it's generated by something else. And there's information processing going on because we know that it does respond to environmental cues. So, how do we characterize that? So, we characterize the movement as accordion like, but there are all sorts of phases of movement within this accordion like movement. And then there are all sorts of movements aside from that. So, the cells can age and break, this colonies can break apart. There are all sorts of behaviors. This is just one small subset of behavior. And so, even within this, we can look at the behavior in terms of this non-neuronal cognition framework. We can model this as a series of coupled oscillators, but the movement can vary due to stimuli. And you'll see what I mean by that in a little bit here. So we talk about non-neuronal cognition. It was defined in Lyon in 2015 in this paper in Frontiers in Microbiology as the total set of mechanisms and processes that underlie information acquisition, storage, processing, utilization. So if you know about the way people kind of view cognition, it's largely as information processing. And so this is consistent with this, but non-neuronal cognition doesn't involve neurons. So what does it involve? Well, in Beluska and Levin in this paper, they talk about different ways, different places you can encode information in terms of non-neuronal cognition. And they point out that the extracellular matrix is ideal as a memory medium. So that's maybe where you get some of the things that are stored in terms of memories or in terms of information and information processing. But they also talk about something called Stigmargy. And this is something from the complexity literature that's kind of obscure, but if you've read about like swarms and other types of collective insect behavior, you've maybe heard this term. Refers to the physical traces that can be referenced and read in the future. So in ant colonies, for example, you have ants moving around and they're navigating a landscape and they're making, they're wandering around trying to find food, and they can form these trails. And the trails are, of course, not, they don't necessarily follow they follow cues, but they do more than that. They lay down pheromone. And then other ants will follow the pheromone of their conspecifics. And eventually, over time, they'll build this trail system. And the trail system, of course, looks like it was organized by a single brain, but it was actually organized by a number of brains. And they relied heavily on this external stimulus or this pheromone that was laid down on the ground as a way to sort of guide them to these resources. So one ant might find a resource somewhere. They'll lay down some pheromone along a trail going to that resource. Other ants will follow and reinforce the trail, and then you'll get these ant trails. And it's collective behavior that, there's a brain generating it, but it doesn't require a brain to maintain it necessarily. So that's what they mean by stigmergy. So this is an example of the extracellular matrix in diatoms. I think this is just kind of like a generic example. These are secreted polymers that completely encase the cell and are responsible for adhesion and interactions with the external environment. These are called mucilage trails and strands. So you have these things that, they're polymers that join the cells together, or they link the cells when they're decoupled, and they provide maybe some sort of memory or some sort of way of mediating those collective behaviors by the individual cells. If you go back and you look at the video that we showed here, you'll see that the cells will stretch out, and then they'll be able to stretch against one another, but they don't break that bond. They're always like sort of touching one another at some point. So the extent of the stretch, for example, you see a little bit of the cell in contact. And so the question is, why doesn't it fly off the end of the cell? Like, does one cell not fly off the end of the other cell and break the colony apart? Well, it's because of these mucilage trails. They're kind of sticking together. And so it's not completely, it's not completely unguided. But you say, okay, that's a physical thing. So, well, we can turn to the sensory world of diatoms and see what they might be taking in as an input in terms of sensory input. So there's the sensory world. You have you might find these diatoms in a marine estuary, or you might find them in a river or a lake, or somewhere in a pelagic environment where there's a lot of biodiversity, but they're sort of in the water there. So if you collect a jar full of water, you can isolate them perhaps. And so, but they experience a lot of things that you would find in that kind of environment, like sunlight, different chemical stimuli like CO2, water shear, of course, because you have different things going on in the water column. And so, a couple of examples in the literature of like the things that they get exposed to. One of them is the elevated levels of CO2 or just fluctuations in CO2. So we know that, like, for example, diatoms sense CO2 and they can sense concentrations of it more specifically, and that affects their growth. And so the growth of the colony, the growth of the cells relies on the CO2 concentration. And so when there's high CO2, for example, it'll reduce grazing pressure and it has an effect on photosynthesis. So in nature, like if they're in a marine estuary, this is important because depending on where they are in the water column or in the ecosystem, they'll experience different levels of CO2. And so this is something that they deal with in the natural world. We also deal with water fluid shear, fluid motion. And so there have been a couple of studies looking at that, especially in terms of perception. So there's decision-making physiology and perception. So we're not just making this up that there's like something going on there. People are talking about this in the literature. And diatoms have sensory systems for detecting and responding to fluid motion, osmotic stress, and iron abundance. So, like think about like that water column again. There's shear going on, of course. There are all sorts of fluid motions, wave action, uh, you know, other types of things. And there are also things like osmotic stress and iron abundance that are also playing a role in what's going on in the water column. And so they can perceive all these things and they can adjust their behavior accordingly. Finally, we know that light is something that they can sense, and you can see in those ecosystems that they exist in that they experience light. It's not trivial. So there's actually light intensity modulates the response of diatom species to ocean acidification. So you get these chemical signals and they get modified by light intensity. And you can imagine seasonality, of course, is a context in which they might experience these kind of fluctuations. Also day-night cycles. So there are a lot of opportunities probably to observe these different things in nature. And of course, we can implement them in the lab artificially as well. So CO2 and light availability affect diatom physiology. And in this paper, they tested 3 light intensities, and they found that low light intensity affects the physiology and distinct physiological traits. They also can localize sources of chemokinetic and chemotactic mobility. So they can they almost do like a pheromone guided search to find mates. And so that goes back to the Stigmargy thing that we were talking about earlier with pheromones and other things. So Susan, you had so infrared light at 1300 nanometers penetrates to a greater depth than other wavelengths. Yeah. So there are a lot of if depending what the light intensity is, there are a lot of things that happen in the natural environment, and so that's good. So those are just a couple examples, and I know we talked a little bit about it in the group, and I was thinking, do this to kind of frame this properly. You need to do a literature search. So we did some literature search on it. So it turns out that there's a lot of stuff that can potentially be varied, and you can look at the how diatoms respond. Now, the question is, of course, how do we model this behavior in an ideal fashion? and then what happens when it's exposed to these different fluctuating environmental factors. So the first thing we have is this, what we call collective pattern generator. And this is a model that is based on a central pattern generator in neuroscience. So here are cells, again, they're moving against a substratum, and the substratum could be another cell, not just the surface. And you end up with this actin-myosin system that works. So it's almost like muscle and has the same elements as muscle. And it uses these adhesive mucilages to move against the substrate. Now just replace the substrate with another cell, and I think you see what's going on with the colony, that these actin-myosin systems are moving against, the cells are moving against one another using those. And so, we'll build our model here around the propulsion of the colony as the mode of movement. We'll build the psychophysics model. We'll talk about oscillatory movements, which will allow us to view this circuit, and then psychophysics models will account for a wide range of behaviors outside of oscillatory movements. So here's a CPG. This is a collective pattern generator, and you see these often in terms of like any sort of pacemaker, biological pacemaker, like a heart or some insect legs. When they move, they're usually governed by one of these CPGs in the nervous system, in the periphery. And so there are a bunch of inhibitory and excitatory connections. They're synapses in this case, but if we take it outside of the neuronal context, they're just connections that are either inhibited or excited. And then our COPG model is basically our colony here, each cell being one of these rods, and then these black dots are dots that connect. These are kind of arbitrary locations of these actin-myosin complexes, and then we can link them with this sort of connectionist framework where each link between the two nodes on different cells are labeled with sort of a weight. And so we can weight these connections to make them either excitatory or inhibitory. So there can be a gain, or there can be like a latency between the two things, and that will govern sort of the movement. So we can use a series of coupled sinusoids to model the ideal COPG. So each of these cells is one of these trajectories, and then we can have these overlapping sinusoids depending on sort of the frequency of movement between the cells. So, like an idealized model would be where you'd have something at like half phase synchronization. So there's a lag between cell 1 and cell 2. But these can be tightened up. You can have quarter phase, or maybe like 10 degrees out of phase, depending on what your depending on environmental effects or what the model system should look like. And then you can also add noise into the signal, so you can simulate discoordination. So if you have overlapping noisy signals, that makes it much harder. So we can just think of like the cell maybe like moving around a little bit, like stochastically in space, and then moving against one another stochastically, which would be what this is. And so we can do a lot of this like numerically, and then plotting out like functions, and we can think of it that way. We can also think of this in terms of like if you have an environmental input to the system, maybe on one end of it, what does the movement look like? If you change the weights, for example, so if I'm putting like light sources into these different nodes, I'm trying to like simulate like light intensity. What does that do to the weights? Does it change the weights, positively or negatively, or what? And this is a little tricky because we don't really know what it should look like in nature, but we can play around with this model and, produce outputs that we maybe expect. And so again, in this case, we would have a stretch phase where the thing would oscillate, and then we could, model different inputs and change the weights, and then actually build like a model of something moving, and like actually see, like maybe make a prediction of what the movement should look like. And so in terms of sensory thresholds, we can look at just noticeable differences. So as we increase or decrease the light intensity here, then what's the least amount of difference in light intensity that you need to observe differences in movement in the colony? Does it affect, when does it start to affect the colony? You can also have a signal to noise ratio. So you can have, you have these forces, but again, you don't know, like, they're forces that are noisy versus forces that are like informative to the organism. Like if it's some shear that exists because maybe the seas are rough, is that different from something that where it's just kind of like um, some other type of shear force that it might experience? Maybe there's a large predator coming nearby, and they don't really bother Basel area, but that might be a signal for something. So, we don't really know why they're responding to these, but we can do this sort of signal noise testing where we look at like maybe patterned forces on the cells versus like some random set of forces, that are kind of like jumbled in a sequence. Like you have a large force and a small force and a large force and a moderate force versus like pulses of some shear force, like almost like waves hitting it. So we can do those sorts of things and look at what the outcome is. So then that was the end of the talk, and I point out Thomas Harbeck's observation of Diatom's site, which is nice. He has a lot of stuff on Diatoms there, on his observations of the colonies. So he's helping us with some of the observations, empirical observations, and then I put in a plug for the DevoWorm groups. We have our links here for DevoLearn and some other sites that we have here, the GitHub repo and the main website. And so that's what that talk was about. I think that'll be something that we can explore later in terms of how to actually write up a paper on this. So we proposed a couple of models, potential models here that might be, informative, maybe not just for diatoms, but for the whole enterprise of, cell collectives or cell colonies and how they respond to different things in nature. So it might be a pretty interesting paper to approach it that way. It was actually a pretty well received talk. So, did you have any questions, Susan, or comments?
There, I turned myself back on.
Okay.
Yeah, I'm curious about how big they are and whether I could put them in my new ball microscope and see them, because then you could observe them 3D.
Oh wow, yeah.
They're 3D. Yeah, if the microscopes I've got are, I don't know, you can get 1 millimeter field of view, so I wonder if you could see them in that. That's as narrow focuses I've got currently.
Yeah, let's see. You get like the cells, I think, are 10 to 80 microns, so the colonies and they're stretched out would be in the order of maybe several hundred microns. So I don't know. I mean, that probably would be a good size for that. I mean, I don't know how. Yeah, you'd have to. What I understand, they're kind of hard to work with. Like the culturing isn't too bad. You can actually culture them, but then like working with them is a little tricky. So.
Oh, how do you culture them?
Well, I mean, you go and you get a sample of them, and I think you can get them in like a river or like maybe a brackish water source or somewhere like that, and you just collect them, like collect a water sample, and you look for these colonies, and then you can isolate them into a culture dish. And the culture, I think, is there's some nutrient mix that they use. These, but I'm not really sure. And then like they don't. It's easy to kind of culture them and get them in, keep them going for a while, but then like to maintain them is the harder part.
Okay.
Yeah.
I've been told the best nutrient mix for a diatom is to take some soil and drip water through it so that you've got soil-based water and use that. So I do know something about that, but I don't know if these basilaria occur naturally in Canadian cold water.
I don't know. I mean, like, I think they probably do. Maybe in the summertime, not in the wintertime. But I know like Germany, they have them in Florida, so they're kind of like C. elegans, where you find them all over, um, and they're in the soil. And like you can, I did C. elegans. I've isolated some C. elegans here where I live, and I know you can do it all over the world, and you can find different like um, wild type strains all over the world, like a lot of stuff that's not been characterized. So that's an issue. Like you don't really know what the exact with Basilaria. It's just basically with it's like basically a genus. They have species divisions, but they're a little tricky to define. But I mean, they shouldn't be too hard to find. The question is like I think identifying the right diatoms that maybe you want to look at and maintaining them in culture. So
Okay, how do you find C. elegans in your water?
well, you can find C. elegans. You can harvest them from like a compost pile if you have that, or like the soil somewhere where it's like cool and wet, like in an area that's wooded. So you go to the soil and you take a sample of the soil and um, you put it in a culture dish, like an agar plate, just little samples of soil, and you pack them down. Then you might store them that way overnight, and then you pull up the soil, and what's left on the agar plate is a bunch of things that have crawled out of the soil onto the agar, and some of those things will be C. elegans or some other nematode. And so then you have to take like a worm, what they call worm pick, which is a little metal thing, and you have to pick them out of the agar plate and put them in a new plate with food, which is like a medium of bacteria. And then you can let them sort of, isolate them on those plates. And then you end up with a bunch of nematodes, which you don't know which one is C. elegans. The ones that are C. elegans will self reproduce. And so if you put a C. elegans in a plate on its own with food, it'll proliferate quite quickly in like a day or two. You'll get a population.
Neat.
Yeah, and I have a protocol for this on the website, so it's like it's a nice thing. A nice thing to do for like someone who's trying to learn biology and how to isolate like different things and put them under the microscope. Um, but it's definitely not too hard. I mean, it's hard, but it's not like so difficult. It's not like a molecular protocol. But yeah, I did that. I did that once, and I wrote it up, and it's pretty good. Yeah, so you can observe them, and um, that's why they use C. elegans as a model organism because you can find it everywhere. So,
Where do you find a worm pick?
oh, people make them usually like they'll take like one of these glass pipettes, and they break the tip off, and then that leaves you a little hole at the end of the fat part of the glass pipette, and then you take a piece of metal or little piece of metal wire, and you stick it in there, and you kind of close it up with under using a Bunsen burner. You close it up, you melt the glass around it, and then you like tap the other end of that glass wire or the metal wire so that the end is flat. With like a hammer, you tap the end of that with a hammer, and it becomes flat. And then you have something that you can scoop up nematodes from a agar surface with. You go under a microscope, you take it, and you take the metal into the metal pick, and it's flat now, so it'll scoop up your worms, and they just scoop onto it. You have to. There's a technique to it, so it's not like it's easy. It's kind of tricky to get the technique, or else it'll dig into the agar. But you just scoop it up, put it into a new plate.
That sounds like a really nice science project, perhaps with my granddaughter or something.
Yeah, Yeah.
If she was to homeschool again, we could do it.
Yeah. So,
We're headed for that here.
yeah.
We're going to shut everything down again.
Oh, yeah. Yeah. Well, so that's psychophysics of non-neuronal cognition. Next thing, I guess I was going to talk. I was hoping Krishna would make it to the meeting so we could talk about this paper that he's writing. So he's writing this paper, Reinforcement Learning Algorithms, SARSA and Expected SARSA. So this is something he wrote. He's kind of in the process of writing it. It's on reinforcement learning, which is a particular learning approach that is sort of based on like behavioral psychology, but it's basically where you give the algorithm a reward for doing something that you want it to do, and then it'll learn, like it'll take different policy, it'll choose from different policies, which are ways to be rewarded for certain behaviors, and it'll end up learning things in this way. And so reinforcement learning focuses on the sequence of decisions to maximize a reward, which encodes the success of the outcome of an act and achieve a goal in an uncertain, potentially complex environment. So we talked about reinforcement learning in one of the DevoWorm ML lectures. There's a lecture on it. And so he's writing this paper on temporal difference learning approaches, which is something that we didn't really talk about in the bacillary or the diatom talk, but this is something that you find in a lot of maybe biological nervous systems, and they've tried to model it in artificial neural networks. So it's basic. It's how it's described. So temporal difference learning is where you look at like a sequence of quantities and you try to sort of extract a trend out of that sequence by looking at the differences between the um, the selection of things that you presented with. So if you had, like, for example, a series of days where it either rained or it didn't rain, and you observed those days and you said, all right, it rained today, but it's not going to rain tomorrow, and then it rained the next day, it won't rain the next day. So what should I expect for the future? Can you assemble like a forecast of when it should rain or when it shouldn't rain? So, as you can imagine, that's a little bit difficult to do, but you can do some sort of difference learning on that temporal sequence to sort of get information about the intervals and other things that information you might need to know to make a better prediction. And so you do this over time, you revise your sort of model of what uh, you know what you should expect, and it should bring you nearer to the prediction, a successful prediction. That's the idea. So this is what he's going to talk about, this method, and this is of course your um, reinforcement learning model. You have an agent, and it interacts with its environment through an action. So the agent engages the environment by observing it, maybe, and then there's a state that it observes and a reward for doing something in that environment. So it observes the world and it either makes some decision. So one way to think about this is like with rats with food pellets, because I think that's how they really started looking at behaviorism. They, would have a rat and it would be presented with maybe a food pellet, and there would be like another stimulus associated with it. So, there might be a tone when there's a food pellet. And so, every time there's a tone with a food pellet, the rat either, selects the pellet or it doesn't. And then there's the state of, whether it's been rewarded or not, and there's a reward. It may not be rewarded, or it may be rewarded for doing some sort of behavior. And that's how it works. I know I'm not really that wasn't the most elegant description of that, but, in reinforcement learning, they use a lot of different terminology for this. So, so this is.
I've heard of it before, and it's also how it also gets into robotics, and how robots behave. And there's I love the term epsilon greedy as part of the interesting language that goes with it.
Yeah, yeah, they've developed quite a um, terminology for it. So, and that's a whole nother thing. But this is going to be a nice paper when it's finished. I'm not sure when we're working on it. So I don't know like how it applies necessarily to what we're doing directly in the group, but it's something that is sort of part of the machine learning um, mission here. So it'd be interesting to see what he does with it. As for reinforcement learning, we've talked about this in DevoWorm ML a bit. So if you're interested in sort of what people are doing in reinforcement learning, there's some lectures there to look at. There's also things online that we've talked about that they give you tutorials in terms of like how to implement it in a machine or in a robot, especially. So there's that. I just wanted to also remind us that this, and mainly this is for myself, that this Journal of Open Source Science article is still outstanding, and I have not had a chance to really kind of work on it because I've been busy with Neuromatch and some other things. So this is the next sort of one of the next things I need to do. And this is just again going through. This was from last summer's from the DevoLearn implementation. So this is the actual program, and we're going to be working on. I just want to add a couple more things to it. So it's probably even. I'm just kind of holding things up here. I need to get on it and finish it off. I think the thing I was thinking of doing was to kind of give maybe a little bit more information about like how it works and how it's integrated into this larger DevoLearn systems or this DevoLearn effort. So I'm going to do that. Also, I found this interesting bibliography. It's on evolution of reaction norms. So now we're shifting back to biology more. So this is a nice bibliography here where we have some. It's so this the way this works is you have a bibliography where you have an introduction, you have a general overview of different books on the topic, and then you have all these key citations. So this is almost like a annotated bibliography, and I did one of these when I was an undergraduate, where it's like a long list of references, but you try to annotate them with like introductions and general overview, so you orient the person to the topic. If people are interested in doing this, by the way, I think this is something we can do in this group. We certainly talk about references all the time, so it wouldn't be that hard to do an annotated bibliography, or at least start it. So we could have GitHub has wikis, and so we could actually do this with a wiki on DevoLearn. We could have different topics that are maybe the developmental biology, but they're like kind of computational in nature. They could be like complexity measures, biological physics, things like that. And we talk about these topics a lot in the group. What we could do is just have like a reference list and then make an annotated bibliography where we just simply write up a little bit of introduction on it and a general overview of different books, which would just be like, to orient the reader to like, okay, if you want to learn about this topic more, here's some major themes and here's some books. So in this topic, which I actually like this topic, we haven't talked about it that much in the group. They talk about reaction norms, which are these reaction norms are a means of conceptually, graphically, and mathematically describing this total variance of genetic variability, environmental variability, and the interaction of them, as a powerful tool for decomposing it into its constituent parts, nature, nurture, and critically their interaction. So reaction norms are these things that when an organism is interacting with its environment, oftentimes it'll experience some environmental stress. And you can look at this in terms of like the genotype. So like in C. elegans, you have defined genotypes. If you give one of those, you take a couple of defined genotypes and you subject them all to the same environmental pressure. And we've done this in larval C. elegans. Like there's a starvation response in L1 in the larval stage of C. elegans that if you starve the worm in this L1 period, it will retard its growth, so it won't grow. But then after you take away the starvation, it'll continue to grow. And depending on the genotype, the growth is either, it either starts back at a linear rate of growth, or it starts back at a suppressed rate of growth, or sometimes it'll try to make up for lost time and have an enhanced rate of growth. And so you can measure this by like looking at, inducing this environmental stress on the organism and taking it away. And then given that the genotype, but that they're different, you can watch the different responses and actually characterize them. So I talked about this I gave a talk to the virtual worm group this last spring, and I talked about this work in doing this in C. elegans. So if you're interested, I can send you that link. But reaction worms are a very powerful way, I think, of dealing with this issue of sort of what is environmental plasticity versus what is, encoded sort of in the genome. And then when you have a defined genotype, you can actually, understand this very clearly and, and very concretely. So, how do you characterize a genotypic component versus an environmental component? That's what we're asking. And so there's a whole literature on reaction norms and how they behave in development. Reaction norms are typically described as consisting of an intercept, elevation, or offset. So they usually look at a reaction norm in terms of a bivariate graph, which describes the mean trait value across all environments, a slope, which quantifies the degree of trait plasticity, and 3, the shape of curvature, which is the shape of the function. So you can have a linear function, you can have a quadratic function, or you can have a monotonic function. So the idea would be that when you have this sort of uniform rate of growth maybe in your organisms across genotypes when they're not subject to this environmental stress. Then when you induce the environmental stress, there's this, it sort of suppresses, or maybe it doesn't even suppress the organism. It could be that the organism doesn't respond to it at all. But then there's a response coming away from that, which is, it could be like making up for lost time, which would be some sort of exponential growth. It could be, linear, which means it just continues its growth, but it doesn't, but it is affected by that. So it's basically, it suppresses growth, but after you take away the stress, growth continues linearly, or just totally destroys the growth potential of the organism altogether. And C. elegans, they're different depending on your mutant. I can report that there is that difference. And so, basically the defined mutation is, some functional aspect of the organism. It could be metabolism. It could be, some sort of other developmental thing that you're taking away. And so then that has an effect on how the genotype responds to environment. So it's a really interesting topic in and of itself. And so this actually, another word on this is that I know some of the people in the group are interested in plasticity. And so this is actually very tightly to plasticity and understanding how that works in a model system. So, if you want to talk about, developmental plasticity, for example, reaction norms are a part of that. Understanding sort of the plasticity of an organism. So that's that. If you're interested more, we can revisit this. Also, there are a couple papers. I'll probably just kind of go over a couple of them, or a couple things I want to show before we go. So this is an interesting video here. Why things keep evolving into crabs.
Around 10 million years ago, off the coast of what's now New Zealand, a spiny, leggy creature with claws cruised the Pacific Ocean looking for food.
So this is about like early organisms before the Cambrian explosion, or maybe right around that same time. Why they sort of kept evolving into crabs. And so you had these like different shapes of things that were existing in the ocean at the time, and why did they keep evolving into crabs? And the basic argument here is that there is this shape economy of shape. So crab is like what they refer to as crabs is like a circular or a spherical type phenotype. And so, why instead of like evolving this long uh, phenotype that they keep evolving into crabs? It's kind of an interesting video, and I have it in this folder. I'm not going to play the whole thing because it's quite long, but it's an interesting question. I don't know.
Okay.
What was that?
Will you put that up in the notes for the session?
Yeah, in fact, I can put it in the chat. You should be able to access this folder.
Okay.
Yeah.
I know that crabs are radial development.
Yeah.
Just like starfish. Instead of axial development. So I guess that's the question. Why have you got radial developers and axial developers?
Actually, I don't know if they're thinking about it that way. I think they mean like the basic shape of the phenotype. Like why, but I don't know. I didn't really. It's kind of not at the level of like. It's kind of at a very popular science level, but actually, that's a interesting question about radial and axial development. Which I mean, that's a question of like body plans and why do you have certain types of development, and there's an interesting question in there though. Like why do you have radial development? Why do you get axial development? And
what causes that?
yeah. Like in one of our updates, or I guess one of the things that for the paper, the Boring Billion paper that we're working on, Dick was asking like, why do you have what kind of things were living like in that Boring Billion period? This is before the Cambrian explosion, but after sort of the I guess the last eukaryotic common ancestor, I think. So what is going on in that period of time? That is, what do the organisms look like? And the question is, I don't really know. I don't know if people know, but we can kind of look at the phylogeny and take a guess. And then, again, related to that is why would you get like these different types of development that are just kind of like emerging? Why would they emerge? And because they're obviously emerged very far back in phylogeny, because they're sort of some of the basic modes of development. I mean, you don't have like there, you could imagine that there might be some really exotic modes of development that haven't been tried. Why didn't we get those?
Yeah, why did they die out?
Yeah.
And when did backbones happen?
Right.
Vertebrae.
Yeah. Vertebrae. So yeah, you have notochords that precede vertebrae, but it's like why do you get that axial develop or organization? And it's
because of the notochord probably, but that's a guess.
Yeah. So yeah, that's an interesting. I mean, it's just kind of one of those things that I ran across. I thought it was interesting for the group. There's a paper that you might want to check out. This is Evolving L Systems in a Competitive Environment. So the L systems, or something called Lindemeyer systems, are interesting. Like they're computational models. They're basically where you have this. It's a symbolic system where you can put in different. You can have it's like an agent-based model, and so you repeatedly apply rules to some initial condition, and you watch the growth. So it's kind of like the models that we saw earlier, um, the diffusion limited aggregation models, except it's using a little bit different methodology. It's not using a random like things attracted to other things and sticking together. This is where you have an initial condition, and then you apply production rules sort of recursively, and then you watch the thing grow. And because it's a symbolic system, they're not random rules. They're almost like grammar rules, and so you can build visualizations out of this. And they use Lindemeyer systems a lot to model plant growth. So like you can build root systems and branching systems using L systems. So this paper kind of talks about using L systems in a competitive environment. So this is an example of a simulation where they look at the importance of competition. So again, they're modeling these little plant systems, and they actually can replicate some stages of the evolutionary process. So they can look at leafless agents. They can look at different types of agents producing multiple seeds. So you can start off at a very low level of complexity and build up to agents that are more complex. And so this is an environment. This is an environment resulting from a simulation with a high growth density factor. So again, you can play around with the parameters, and you can get variation. Now you get an ecosystem where some agents try to escape the canopy to avoid occlusion. So the plants are trying to compete for sunlight in this case, and so it kind of drives a lot of interesting dynamics. So agents that develop early on in a simulation are often small and vertical. And so this shows a small colony of low-growing plants. And then after this stage, it gets more complex, then competition starts to dominate. And you get this competition between, different types of agents. So some of these agents are brown spheres. Some of them are these long, tall green things. And so they compete for tallness, basically. They become taller as the competitive process moves on. And that's because they're competing for sunlight, because there are more of them, and it's a more complex ecosystem. So this is a nice, it's just a nice set of different simulations that they've implemented, and then they've done a lot of nice visualization here. So again, they look at leaves and the development of leaves, where they kind of conflate development and evolution here, because you're like developing things that are going to be competitive, and then they evolve. So, but I think they get some pretty good results out of this. And so this agent has evolved in an environment where the light shines only directly down. So in this case here, you're getting sunlight from one direction only. And so that affects the way it grows. And that's interesting in light of the stuff that we were talking about with diatoms, is that you can do this with light sources. You can simulate a colony or something and how it responds to light. So you can actually play with the light sources quite creatively to see what happens to the behavior. And so this is, again, they're looking at they're actually modeling their evolved agents it's based on some illustration of different plants. So they're basing it on a real biological set of examples. And there's the appendix with all the technical details. So again, this is something that you can look at later if you're interested. And then there are a couple other papers I'm not going to talk about. There's this one that was this paper that I think Dick sent me on from molecular systems biology. This is Deep Cycle. So this is something that reconstructs a cyclic cell cycle trajectory from unsegmented cell images using convolutional neural networks. So this is something that we've looked at doing. We've done a lot of stuff with analyzing cells in this group with machine learning and with deep learning. And so what they're doing here is they're using unsegmented cell images to reconstruct cell cycle trajectory. So they're using convolutional neural networks, which is something we've used in the group. So they're basically, so the abstract reads, the advent of single cell methods is paving the way for an in-depth understanding of the cell cycle with unprecedented detail. And so we need to evaluate the cell cycle progression because it's critical for an exhaustive cellular characterization. Here they present Deep Cycle, which is a deep learning method for estimating a cell cycle trajectory from unsegmented single cell microscopy images. This relies exclusively on the bright field and nuclei-specific fluorescent signals. Deep Cycle was evaluated on 2.6 million single cell microscopy images. And so what they did is they visualized different cell and molecular components that are involved in cell cycle using fluorescent markers. And so those fluorescent markers then retract, and they were able to sort of characterize cell division and the cell cycle. So the cell cycle is this very specific thing in biology where a cell will go from a single cell to two cells. And so this involves a process of division and pulling apart the chromatin and forming two different cells as opposed to a single cell. And so there's this whole process. There's also a molecular process, like a circuit where the cell goes through different phases of replication, and they're very specific. There is a very specific cycle of things that have to be turned on and shut off at different times to make this work. And so they're able to look at these live cell images and look at these fluorescent markers, and from that sort of build a model of cell cycle. So
wow, if they want to do that some more, I could do that with the axolotl eggs.
Yeah. So they validated the deep cycle trajectories by showing its nearly perfect correlation with real-time measured images. So they were looking at real-time images, and they used those real-time measurements, and they correlated what they did with deep cycle. And then this is the first model that resolved the closed cell cycle trajectory, including cell division, solely based on unsegmented microscopy images. So they didn't have to segment the cells, they just had to follow the fluorescent markers, and they were able to do this. So this is the way they sort of took a bright field image with host staining, and they put it into this model. And then they were able to derive a classification score, and then they were able to do this and build these. And this is we talked about UMAP a couple weeks ago. They use a UMAP method to sort of characterize all of the different phases of cell cycle and where they lie in the space. And so they're able to separate out G0 and G1 from G2 and M. And that's basically how they did it. Without getting into too much technical detail. But if you want to read this paper, it's in the folder here. So yeah, any thoughts about that, Susan, other than the axolotl example?
Well, I'm hoping to build this spherical microscope, and if one could buy more expensive small microscopes like they were exhibiting in Neuromatch to go on top of a mouse's brain, if you had those, then you could do fluorescent imaging of the whole surface of the egg dividing. And initially, at least up until, 6 to 8 cell divisions, that's what you're looking at is simply the cell divisions that are happening. Like there's nothing happening underneath until a little later. So very early on in development, you're just seeing all of the cells that are dividing.
Yep.
I don't know if that, I haven't studied any other animals in any depth. So I know that Mammals are very difficult to study because they need to be in a special environment, high CO2 and warm, so they make special stages that are worth a lot of money in order to sell, to discover what's going on with mice and rat embryos. And I'm sort of sticking with the amphibians because they're cheap and easy. Oops, pardon me.
Okay. Okay, well that's good. We'll think about that some more. I think that's an interesting approach and topic. We've often thought about, like, when we segment embryo images, we have to characterize cell division, and doing it by segmenting cells only gives you so much information to go on. So you can see, like, if it's dividing, but then that's also a challenge when you're just trying to segment like images of the cells with their membrane, and trying to segment out the membrane is when they're in the process of division. That's going to break the algorithm because it doesn't have the ability to. I mean, you can train it on cells that are dividing, but it's kind of hard to know. It can break the algorithm in terms of, like, is this a cell or not, or are these two separate cells? Or so I like the way they do this with markers of cell division and being able to predict. But that being said, it's not exactly what we want to do with embryos, but yeah, I think it's a step towards understanding that. It's really good. So again, that's all in that folder. I can make that folder available to people if they want to email me, or if Susan has the link there, and it should be shareable. Otherwise, I think that's it for today. Thanks, Susan, for attending. And so we just changed our time. So we had to change the UTC time of the meeting, and hopefully people can make it in subsequent weeks. I know the last couple of weeks have been a little rough because we had things going on, and but I think next week we'll have an update on maybe some projects. I think Ojwal might be able to attend next week and give an update on actually, I think he's been doing some things on the axolotl microscopy images that he has an update on, but he hasn't had time to make the meeting. So.
Okay, I may have some more different images for him at some point here, hopefully.
Okay. All right. So thanks again for attending, and have a good week.
Yes, bye. You have a good week as well.
Too. Thank you. Bye.
Bye.